by bacterial hyaluronan synthase. Okay.
Hi, my name is Mehul, and I've been working for the past year and a half in the Zimmer lab on the translocation of hyaluronan by the bacterial hyaluronan synthase. And so what is hyaluronan? Hyaluronan is a extracellular polysaccharide made of N-acetylglucosamine and glucuronic acid. It composes a major part of the extracellular matrix of most vertebrate tissue and affects such processes as wound healing, cell proliferation and differentiation, embryological development, and angiogenesis. It's also used by many pathogens such as Streptococci species to encapsulate themselves in a coating to prevent to escape immune response. It's also overexpressed on many cancer cells for anchorage-independent growth. So we know a lot about how or the effects of hyaluronan, but we don't know much about how it's actually made. We know that it's made by the hyaluronan synthase on the inside of the cell, but we don't know how it gets to the outside of the cell where it performs all of its functions. So that's where my project comes in. So there are two dominant theories to how hyaluronan is translocated. The first incorporates the use of it synthesizes hyaluronan as it translocates it at the same time. The other incorporates the use of an ABC transporter, which tightly couples synthesis and translocation. So the first way we approached this question was we wanted to engineer E. coli to become efficient producers of hyaluronan. And the way we did this was we had to manipulate the pentose phosphate pathway. Now, E. coli don't normally produce hyaluronan. And the limiting reagent in this reaction is the UDP-activated glucuronic acid. So we needed to upregulate this molecule. The way we did this was we inserted the GAL-U and GAL-F genes, and they encode for the enzyme that converts glucose 1-phosphate to UDP glucose. We also inserted the UGD gene, which encodes the enzyme that converts UDP glucose to UDP glucuronic acid. And then we inserted the HOS-A gene from Streptococci species, which takes the UDP-activated sugars and produces hyaluronan. So we wanted to grow these cells up and ask the question of where are they located? Well, where can the hyaluronan be found? Now, there's four possibilities. It can either be found in the medium, on the surface of the cells as a capsule, in the periplasm, or inside the cells. Now, if there is no translocation, we'd expect it to be inside the cell, obviously. But if there is translocation, we'd expect it to be found in the periplasm. This is because our hyaluronan synthase is from a gram-positive species, so it wouldn't have the machinery necessary to translocate across the outer membrane. So the way we did this was we set up 4 samples of the medium, the intact cells, spheroblasts, which are gram-negative cells with the outer membrane degraded, and the whole cell extract, which are just lysed cells. And we measured the amount of HA, or hyaluronan, using an ELISA assay, which uses a hyaluronan binding protein attached to a horseradish peroxidase. And when it binds hyaluronan, it emits a color that absorbs at 450 nanometers. So we wanted to check this for 3 different cell strains that we created. The first one's a PET28A vector that lacked the hyaluronan synthase. So this is our negative control, and it shows no activity, as expected. Then we tried this with the Streptococcus equisimilis hyaluronan synthase. And this synthase is extremely active, and as seen, there's saturating levels of activity in all the samples. And we believe this is because there's so much hyaluronan in the sample, in the cell, that it actually caused the cell to break open and be released into all samples. Now, when we look at the Streptococcus pyogenes, we see a pattern that we would expect with translocation. So there's a lot of activity in the whole cell extract sample, and then there's a small amount of activity in the spheroblasts, more than the cells in the medium sample, though. And we believe here that there was translocation into the periplasm, but we don't have equal levels of these 2 activities because there was incomplete spheroblast preparation in that the outer membrane wasn't degraded completely, so not all the hyaluronan was available to be bound to by the hyaluronan binding protein. To confirm this, we wanted to look at these cells with electron microscopy. So when we imaged these cells, we actually found that compared to normal cells, these cells were actually, many of them were actually dead, but a lot of them had these protrusions right here of electron dense mass. We think that's hyaluronan, and what I believe happened is so much hyaluronan accumulated in the periplasm that the outer membrane actually broke and started leaking out hyaluronan. right here. So at this point, we believe that the hyaluronan is translocated by the hyaluronan synthase. But we wanted to make sure of this by looking at the system in vitro. We move on to an in vitro system by looking at inverted membrane vesicles. What they are is we took E. coli cells expressing the hyaluronan synthase. We grew them up and lysed them, and we applied the membrane fractions to a sucrose cushion. Now, when you lyse the cells, they're going to spontaneously reform vesicles where the proteins in the membrane are in an inverted orientation. So now, after we isolated them, the proteins are now, instead of facing the outside, they're facing the inside. And they contain all sorts of the E. coli proteins, but we have our hyaluronan synthase overexpressed in them. So then we developed an assay to test for translocation here. So here's our inverted membrane vesicles, and if there is when we add the UDP-activated sugars, if there is translocation, it will move inside the vesicle because it's inverted. And if there is no translocation, it'll be found on the outside. Now we can test for this by adding a hyaluronan-specific degrading enzyme called hyaluronidase. When we add that, only the polymer on the outside will get degraded. Everything on the inside will be protected from degradation. Then we can add detergent to solubilize the vesicles and degrade everything. And we can test for translocation. If we see a large difference between these two samples, we know that there was protection, so there was translocation. If there is not a big difference, then there was no translocation. The way we detected this is we added tritium-labeled UDP and acetylglucosamine, and then we could just measure the radioactivity with scintillation counting. So but first, we wanted to test whether these IMVs actually could make hyaluronan. So we did a time course of 10, 30, and 60 minutes and found that there was a progressive increase in hyaluronan production. Now, we did some controls. When we added a detergent, Triton X100, and the hydrolase, we got down to background levels, so it was degradable. Then when we didn't add UDP glucuronic acid, it was also down to background levels, saying that you need both sugars for synthesis. We also made IMVs with an empty vector that lacked the hyaluronan synthase, and there was no activity in this one either, as expected. And then if you add EDTA from the beginning of the reaction, there was no synthesis either. This is because EDTA chelates magnesium, which is a necessary cofactor for hyaluronan synthase. Then we moved on to the assay. We wanted to check a couple things in the assay. First, the question of, is an ABC transporter involved? And if an ABC transporter is involved, there would need to be a constant supply of ATP. So we needed a ATP regeneration system for the reaction mixture. And we created that by adding creatine, phosphokinase, and phosphocreatine. So the phosphate group from the phosphocreatine was given to the ADP to rephosphorylate to make ATP, to keep a constant supply of ATP over the course of our reaction. We also wanted to look at proton motive force. So in bacterial cells, bacteria use NADH to create a proton gradient with H+ being pumped outside the cell. Now, hyaluronan is a highly negatively charged polymer, so its export to the outside of the cell would be facilitated by its quenching the chemical gradient. So we believe that proton motive force could actually help export of hyaluronan. But when we looked at this, we saw 50% of it translocated in the normal assay, and when we created a PMF or a constant ATP concentration, we didn't see much of a difference, or with both of them, we didn't see much of a difference. So here we're not sure. We don't think that an ABC transporter is involved, and we think it's the hyaluronan synthase. But from here, we wanted to be sure that it was a synthase. So what we did was we purified the hyaluronan synthase and put them back into proteoliposomes. So we took hyaluronan synthase, put it into detergent, added it into detergent solubilized lipids, and when we removed the detergent, they spontaneously reformed lipid vesicles. So now, just like the IMVs, the hyaluronan synthase is pointing in towards the inside of the vesicle, but now it's the only protein inside of this vesicle. So we wanted to make sure they were active, and when we did a time course, the activity increased linearly. We can also degrade it, and without the glucuronic acid, it wasn't made. So back to this assay, we wanted to see a large difference between these two conditions. And this is precisely what we see. We see almost 80% translocated here, but in this case, everything's degraded. So the hyaluron synthase must have been efficient at translocating hyaluron into these vesicles. So my conclusions from this project are neither a proton motor force nor a constant ATP concentration are needed for translocation, which indicates that ABC transporter is not really involved in this system. Also, that the hyaluron synthase can be purified and is active in proteoliposomes, and the hyaluron synthase is sufficient for synthesis and translocation, suggesting that they're tightly coupled processes. Now, this is important because the hyaluron synthase shares homology to the synthases for many other major polysaccharides, such as the cellulose and alginate synthases, and they likely share the same method of translocation. Additionally, this is a novel mechanism in biology. Most major macromolecules, proteins and DNA and RNA, they're exported out of the nucleus or into the endoplasmic reticulum by a different set of enzymes that actually make them. Here we see that the hyaluron is actually made and exported by the same enzyme, which we haven't really seen much of in biology. In the future, we'd like to look at how exactly the hyaluron is translocated and synthesized, and we'd like to determine how the hyaluron synthase determines when to terminate polymerization, because hyaluron's effects are mediated by its length, so it'd be important to know how it regulates this length. I'd like to thank Caitlin for all of her work on the proteoliposome assays. I'd like to thank Olga for her help in the lab, Dr. Zimmer for helping me through this project, and the Harrison Institute for funding, and you guys for being here. Thank you.
In the inverted liposome math, you showed a time course.
Yes.
How could you get time zero? How did you initiate the reaction? And then why couldn't you use this as a corresponding method to show a time course in the intact E. coli, where you show that they just burst? Well, could you generate a time course there as well?
Well, the difference in these two things is we're measuring radioactivity in here, and we can't add radioactive nucleotides to those E. coli cells. They're using nucleotides that they've already made by themselves to synthesize the hyaluron, whereas we add commercially available nucleotides.
That's T0.
Yes, as soon as we add the nucleotides, that's when we mark the beginning of the reaction.
Where's the energy that's coming from? You're transporting against the concentration rate.
The hydrolysis of the UDP sugars off of the UDP-activated N-acetylglucosamine and glucuronic acid.
Questions? Thank you.
Thank you.